Okay guys, now let's talk about genetic tools, which is a very important topic. Okay, so what is genetic tools? Means how we can edit our genome. That is genetic tools. So normally the DNA will give rise to RNA and RNA will give rise to proteins. Okay, so now you can do two things. What are the two things you can do? So either you can directly cut the DNA itself. So if you cut the DNA itself, this is what is called as knockout technology. What is this called as? Knockout technology. Or what you can do is either you cut the RNA or you decrease the protein synthesis. Okay, so whenever you change the RNA expression, this is called knockdown technology. So basically genes can be altered, either you cut the DNA or you alter the RNA. So knockout technology is permanent modification. Obviously, if you change the DNA, then it is not going to revert, isn't it? So this is done by a molecule called as CRISPR. Whereas when you change the RNA, you have not cut the DNA. So this is a temporary modification, and this is usually done by microRNAs. Okay, so this is usually done by microRNA. So microRNA usually do knockdown technology. CRISPR does knockout technology. So let's understand what are they and how do they work. First, let's talk about knockout, which is a temporary modification. Okay, so let us see how does a knockdown technology works. See, knockdown technology usually works with the help of double-stranded RNA. So this double-stranded RNA is usually a hairpin shaped, hairpin looped RNA. So this RNA is basically your pre-microRNA, which is formed in the nucleus. And when it is exported out of the nucleus, this is exported with the help of a molecule called XPO5. Now, once this double-stranded RNA comes out into the cytoplasm, this is immediately acted upon by a molecule called as dicer, which breaks this double-stranded into single-stranded microRNA. Now, this single-stranded microRNA doesn't know that where I have to go, what molecule I have to work on. So it needs a CAR. So its CAR is ribosome-induced silencing complex. Okay, so it sits in its CAR. So what is the CAR? Ribosome-induced silencing complex. It sits in the CAR and it goes to the target site. So now let's look at this. Okay, so let's say this is the target RNA. So this is the target mRNA. It's coming. Let's say you have a sickle cell problem. So patient is having sickle cell DNA. It is producing sickle cell RNA, and then sickle cells are coming in the blood and causing problem. So what you can do? You can cut the sickle cell RNA itself. If you cut the sickle cell RNA, and automatically sickle cells will not come in the blood, then there will be no vaso-occlusive crisis, no anemia. So how can you do this? So you can do this with the help of, how you can do this? So you can do this with the help of microRNA. So you cut the RNA. So how will it happen? So you have just now studied from the nucleus, double-stranded microRNA has come up. Dicer has broken this double-stranded into single-stranded. It is sitting in its CAR called as RISC complex. Now this RISC complex will bring the mRNA. So can you see this? So this pink one is your microRNA. It's sitting in the CAR and coming. Now there can be two things which can happen. One, this microRNA can be perfect match to the target mRNA. It could be perfect match. So if it is perfect match, it will cut this mRNA and sickle cell RNA is gone. So you imagine that this mRNA is a sickle cell mRNA and the sickle cell mRNA is cut and it's gone. So now if there is no sickle cell mRNA, there are no sickle cells in the peripheral blood and you're cured of sickle cell disease. Second situation that can happen is that this microRNA is not perfect match to the target mRNA. So it's not a perfect match. So when it's not a perfect match, still it will sit in front of the ribosomes like a stool. Now ribosomes are coming to form proteins, so it will say move, move, but this microRNA with its CAR will not move. So what will happen? The ribosomes will not be able to move forward and make the protein. So can I say that there are two things which can happen due to this procedure? Either you have cut the mRNA. Once you've cut the mRNA, automatically there are no sickle cells. Second, you have caused translation repression because you're sitting in front of the ribosomes, so proteins are not formed. So can I say that microRNAs can have two effects? One, they can break the RNA. Number two, they can suppress the protein synthesis. So the ultimate effect is that the RNA is not able to make proteins. So there is translation suppression or there is mRNA breakdown. The ultimate effect is the gene is silenced. So you have sickle cell DNA. Sickle cell DNA is forming RNA, but RNA is not forming proteins. So your sickle cells are not coming in the peripheral blood, you're cured. Now the problem with this is that you will have to inject, or you have this sickle cell, this microRNA has to be supplied every day. Otherwise, what will happen? The DNA is there, it will form the RNA will form the protein. So these synthetically formed microRNAs are called as siRNA. So they are small intervening RNAs, they are small interfering RNA or small intervening RNAs. They are the ones which are basically small interfering RNA are basically the artificial given microRNAs, which you should be very clear. So now, once you know this, so there are some questions that can be asked. Okay, one, are microRNAs available in your body? Yes. So we have more than 6,000 microRNAs in our body, right? They are responsible for controlling the gene expressions. Okay, so that is why only 1.5% of your genes express, 98.5 doesn't, because these microRNAs control the gene expression. Second, can you have mutations in the genes coding miRNA? Yes, dicer mutations are very common. So if you look at the dicer mutations, so dicer mutations are very, very common. So dicer mutations are commonly seen in unique pediatric tumor, blastomas. Remember, there will be a lot of blastomas which will be coming here. So blastomas, okay, blastomas which will be coming here. So we have pleuropulmonary blastoma, which is again a pediatric tumor, or pituitary blastoma, which are common. Apart from that, this is a very unique tumor and has been asked as a question so many times. This is ovarian sex cord Sertoli cell tumor. So ovarian Sertoli Leydig cell tumors are very classically associated with Sertoli cell, with dicer mutations. So remember this point always. Apart from that, Wilms tumor, multinodular goiter can also occur. So these are the common cancers which can occur due to dicer mutations, which you should be very clear with. Okay. Apart from that, you have to remember. So I told you this is ovarian. Okay, don't never forget that. Okay, dicer. So who dices the sabzi in your house? Okay, so it is ovary. Also females usually. So ovarian Sertoli Leydig cell tumor. Always remember that. Okay. Now, once you're clear with this, once you're very, very clear with this, now that is the function of knockdown technology. Okay. Now let's come to knockout technology. That is your CRISPR. So first thing you should know what is CRISPR. It is called as clustered regularly interspaced short palindromic repeats. So these CRISPR molecules are not found in humans. They are actually found in bacteria. Now if you look at the bacteria, no, he has a lot of problem. What is this? That all the time the bacteria puts its genome into the virus. The virus puts its genome into the bacteria. So viruses put their genome into the bacteria to make them bacteriophage, isn't it? So to prevent this, the bacteria have made this molecule called CRISPR, which changes into gRNA. So this gRNA is again a special RNA. Now it has a Superman car, not a simple car as RISC. This is a Superman car. You know Superman car? If you looked at the Superman car, it can do anything by itself. So this Superman car has a scissors in front. So it can go and directly cause double-stranded DNA breaks. So it breaks the DNA of the virus and the bacteria is cured of the virus. So that is how the bacteria is cured of virus with the help of CRISPR-Cas9 complex. Now, so humans were so excited to see this. Wow, what a beautiful thing. Let's take the gRNA from the bacteria. So what did humans do is they took the gRNA from the bacteria. When they took the gRNA from the bacteria, they made two regions out of it. One was called as constant region, the other was called as variable region. So constant region you can't change. It will work just like bacteria. That means it has to work, it has to bind with Cas9 and it will cause double-stranded breaks. But then we have a variant region. This variant region is the one which we introduced. So now we introduce this to add the new genes. Suppose I'm talking about sickle cell disease, okay? So if I'm talking about sickle cell disease, okay, so sickle cells have a sickle cell gene on the beta. Now you want to cut it, okay? So you used CRISPR to cut it. So immediately what will happen is that this molecule CRISPR this will be cut. Okay, so this double-stranded breaks has happened, and the gRNA is now, this double-stranded DNA breaks have occurred, and the sickle gene is cut. Now, is that enough for you? No. Don't you want to introduce a normal beta gene? Yes. Don't you want this person to make a normal hemoglobin? So for that, we want to introduce the normal beta gene. So this normal beta gene has to be incorporated. So that is done by the variant region. Okay. So now we add this donor DNA, which is attached to the variant region. So look at this. So as soon as you put the donor DNA, this donor DNA will go and insert into the cut ends. Okay, so these are the cut ends. Now in between the cut ends, that donor DNA will go and get incorporated. So once this donor DNA gets incorporated into the DNA, this is what is called as homologous DNA recombination. What is this called as? Homologous DNA recombination. Now this donor DNA becomes a part of your DNA. So this donor DNA becomes a part of your DNA, and now this person will start making normal beta genes. So he'll be perfectly normal now. Now sometimes what happens is, suppose there was a news which was there long back before COVID, that there was a Chinese who did a mutation in the DNA of a newborn child whose parents were infected by HIV. So when he used CRISPR technology, he cut the site where the HIV virus could bind. So that's it. And the child didn't get HIV. So that if you only cut the double-stranded DNA and you don't insert anything, then also what will happen is these two ends, they're non-homologous to each other. So they will just bind together. This is what is called as non-homologous end joining. What is this called as? Non-homologous end joining. Okay, whereas when you incorporate the DNA, you have made the ends homologous. So what is this called? Homologous DNA recombination. Can I say, if you add homologous DNA, then this is homologous DNA recombination. If you do not add homologous DNA, then this is non-homologous end joining. So we have non-homologous end joining, or we have homologous DNA recombination. So is everybody clear with this? So these are the two types of joining that can happen here. Please remember, so non-homologous end joining defect is also seen in SCID. So this defect is also commonly seen in severe combined immunodeficiency. Okay, so this is also commonly seen in severe combined immunodeficiency, and this also has been asked as a question once. Okay, so that should be very clear. So that is how the gRNA works. Okay, so that is how the gRNA works. It causes the double-stranded breaks, and then if you add the homologous DNA, it will cause homologous DNA recombination. If you don't add homologous DNA, it will be non-homologous end joining. So that's a classical pathway of your CRISPR that everybody has to be very clear with, right? So let's look at this question. So gene silencing means gene which will be expressed under normal circumstances will be switched off by the cell machinery. Which of the following cellular component is not involved in gene silencing? So microRNA is a double-stranded RNA. So that is involved in your gene silencing. It is a double-stranded RNA. siRNA is a synthetically made microRNA. A ribosomal RNA is a single-stranded RNA, and it is not involved in gene silencing. So that kind of questions can be asked, and this is about gene editing, which everybody should be very clear. So I hope everybody is clear with knockdown and knockout technology, and you will not do this wrong in your exams, right guys?